NIH, NLM, logo watermark in the top right-hand corner. Title: The Discovery of Novel RNA Viruses in Metatranscriptome Data. An excerpt from a conversation with Eugene Koonin, PhD, November 2022. An image of a page from a research paper with graphic illustrations of the world map, bacteria, and virus. The word cell is on top of the page. Text below it reads, "Expansion of the global RNA virome reveals diverse glades of bacteriophages."
The paper is about the massive discovery of novel RNA viruses in what we call metatranscriptomic data.
Eugene Koonin, PhD, NIH, Distinguished Investigator, NLM, NCBI. He is seated at a table in a large room across from a woman wearing glasses.
In simpler language, metatranscriptomic data is simply the sequences of all RNA molecules that can be isolated from a particular environment. In this way, you get an unbiased sample of the diversity of viruses, and this really opens up a perspective that was completely unavailable before. You discover many novel groups of viruses that have not been seen by anyone before, simply because their hosts are unknown and have not been grown in the laboratory, and accordingly, before the development of this new type of methodology, there was simply no way to see them.
Patricia Flatley Brennan, RN PhD, Director, National Library of Medicine.
We're very concerned here about at NLM about the purity of the sequence samples that we get in. And I wondered if are you able to find the level of purity that you need to understand these different pathways?
Purity is not a simple concept. Purity with respect to what? The way we work here is, in a sense, conservative, because we have certain signatures, certain tags,
okay,
that we know are associated with this type of viruses, and we detect those as best we can. So we try to combine high specificity with high sensitivity. But if something is absolutely novel somehow, unrelated to anything that has been known until now, then we would miss it. Other approaches are necessary, and in this case, the issue of purity and conversely contamination looms very large. The approaches that we have used, at least in this particular project, I don't think so. I think our rate of false positives is very close to zero.
Now, this particular article in Cell introduced the idea that there's more diversity, and there's also more similarity. So how do you balance diversity and similarity? You said there's more than we had expected, but also some similar patterns that we hadn't expected.
There is considered much more diversity than we expected. At the same time, in previous analysis, on a considerably smaller scale, we have, I believe, established the overall structure of this part of the world of viruses, or this part of the biosphere, if you will. Having identified the large division, so-called phyla, and however huge the newly discovered diversity is, most of it does fall into those previously established major divisions. So there is this, I would say, rather satisfying interplay between the already established course structure and the newly discovered diversity.
Text: The Discovery of Novel RNA Viruses in Metatranscriptome Data. An excerpt from a conversation with Eugene Koonin, PhD, November 2022. Gray and blue logo, NIH, NLM, Serving Scientists and Society.